Yeah, and well, they don't have to inject it in anybody to test it. They don't have to look for the illness. All they have to do is look for the protein in the test tube. You know, just look to see if it's producing the protein. Here's the thing to remember about bacteria too. It's the genes that they possess that's the most important thing. You can have several different strains of the same species. There are several different strains of E. coli. You know, there's several different strains of Salmonella. And as we've talked about before, a lot of these have plasmids, extra bits of DNA that give them their pathogenicity, if you will, their virulence. And you can have microbes of the same species, and one of them cause a disease, and the other one not, simply because it has a little bit of extra DNA that it picked up from a plasmid or some other place. So in the case of bacteria, species isn't always important as to whether or not it causes disease. Strain, you know, presence of DNA, what are the proteins that it's producing that's important. Okay, let's turn our attention then to the mechanisms of pathogenicity. How do the microbes cause the illnesses? There's four mechanisms that we're going to mention here, and we've actually talked about each of them a little bit. The first is you can cause illness simply by producing a toxin, production of a toxin that gets ingested. Our examples of this, we've talked about Clostridium botulinum. Produces a toxin that gets ingested. Staphylococcus aureus produces a toxin, gets ingested, causes an illness. Colonization of membranes. Followed by toxin production. Example of this is Vibrio cholera, colonizing the small intestines, producing a toxin that causes the condition. Invasion of host tissue. So now here, you've got to have either trauma to the skin so that the microbes can get into the tissues, or they've got to be able to penetrate past the mucous membrane. A lot of times these microbes that cause this kind of infection have a vector or have some other way of getting introduced into the tissues. Some of the things that you can think of, Yersinia pestis, we talked about this first day of class, causes the Black Plague. Invades host tissues and produces its damage. Salmonella, Mycobacterium tuberculosis, invade host tissues. And then you can invade host tissues and produce a toxin. Streptococcus pyogenes is a microbe that does this. So how do these infections become established? A series of things have to happen. First, most important, the cells have to adhere to to the host. What we see when we look at bacteria is that often on their pili, they'll have molecules known as adhesin molecules. Different than the adhesion molecules we talked about on our endothelial cells that catch macrophages and pull these in. These adhesin molecules, and they're not always on the pili. Sometimes if it's a microbe that has a capsule, they can be associated with the capsule as well. But these adhesin molecules, they serve to bind to glycoproteins that might be on the cell. So if you imagine our cell membrane here, part of the cell membrane are these glycoproteins. Now these glycoproteins, we've talked about them before, these are surface receptors. They're for a specific molecule. You know, and those molecules will adhere to those surface receptors and the cell will respond accordingly, whatever it might be. It might be a cytokine, surface receptor for a cytokine, for a neurotransmitter, for a hormone, any number of molecules. It just turns out that the microbe is taking advantage of these surface receptors. The surface receptors aren't there for the microbe to adhere to. The microbe has evolved a molecule that allows it, if you will, to bind to these glycoproteins. So in this way, the bacterium can adhere to the cell. There is great specificity here. The cells that the bacterium are going to be able to bind to are going to be very limited. Some cells can only bind to, epithelial cells of the lungs. Some will only bind to epithelial cells of the intestine. Yes.
By colonization of membranes, you mean our mucous membrane?
Yes. Yes. Not the cellular membrane. That's a good point. So you have adherence that must take place, and then colonization. Colonization happens after the cell is adhered, and basically what we're talking about is multiplying. The cell has got to multiply if it's going to cause infection. So that means then it's going to have to compete for space, it's going to have to compete for nutrients, it's going to have to overcome any toxic products that might be produced by our normal microbiota. It might have to counter some of the host defenses. One of the things that the host might do is send out antibiotics that bind to the adhesin molecules. Okay? So the host is fighting off this infection. The bacterium, in its efforts to thwart the host, can shed these pili, getting rid of the antibiotics that are coating it. Now the macrophage won't recognize it as being something to ingest because the antibiotics are there and it will ignore it. So in this way it can avoid some of the host defenses. The other thing that it can do is that it can change the antigenic properties on the cell membrane. So if this antibody is binding to this particular molecule, maybe it changes this molecule to something else, and so now the antibody can't bind. So there's antigenic variation that some of these microbes employ to help them get past host defenses. In addition to that, some of them produce proteases. These are chemicals that break down proteins like antibodies such that they can't bind to the bacterium now. So several mechanisms that allow it to multiply. And you have to imagine that really is necessary if the infection is going to take hold. A few cells generally aren't going to cause you an illness. So if you have a microbe that's adhered to a cell membrane and they start to divide and maybe there's 5 or 10 or 20 of them and your immune system shows up, it probably can take care of those 20 cells and you never even know that you've had a minor infection going on. But when they have defense mechanisms like I've just described here, a lot of times that gives them several days to multiply before the immune system can respond to those defense mechanisms.
Did you say that what did you say about proteases? You said some produce chemicals.
Yeah, these are enzymes. Proteases then are enzymes that the microbe produces that destroy host products, so antibodies, complement, things like that.
So, you know, in previous, we learned antigenic shift and drift, and are those related to proteases?
No, because the antigenic drift and shift apply to viruses. Viruses aren't going to be producing any kind of chemicals. So you have adherence, you have colonization. The last thing that has to happen is delivery of effector molecule. Now here, what we're talking about is the molecule that actually does the damage. Now for you to have an infection, you have to have damage to cells. Now this isn't to say that all microbes are going to be producing an effector molecule. You know, for instance, those that just invade host tissues, they may not be producing anything that needs to be delivered. Now, they may just simply be growing and their presence there be the infection, causing disruption of cell. But in most cases, there's something that's got to be delivered to the cell, either to cause the microbe to be taken up or to deliver a chemical to cause water to be lost. In many cases, the microbes are using what is called a type 3 secretion system. And a type 3 secretion system really is like using a hypodermic needle to inject this delivery, or to deliver this effector molecule to the host cell. Sometimes they're called an injectosome. And we see that a lot of gram-negative bacteria have this type of delivery system where they essentially adhere to the cell and then inject the delivery molecule directly into the cell. So they puncture past the membrane.
So it's kind of similar how the virus, or like how they inject.
Similar, but not quite. Yeah.
Like the part that stays outside the cell, same idea, right? Injections.
Yes. Real quickly, if we talk about invasion, when we think about the anatomical barriers, the skin, in order to penetrate it, I've mentioned before, you're going to have some kind of trauma that takes place. But the mucous membranes, not so. What we see with the mucous membranes, and this is the most common route of infection, is that some bacteria take advantage of the systems that have been employed to sample the normal microbiota, if you will. Some microbes can cause direct uptake. So if you imagine we've got several cells lining our mucous membranes here, and if you're familiar, a lot of our cells have these microvilli. Well, those microbes that are able to get past this mucous membrane usually adhere to one of these microvilli, deliver an effector molecule that causes the cell to lose its microvilli and take up through endocytosis the microbe. So through direct uptake,